The steps of a muscle contraction by Sam, Liv, MJ, and myself, Sydney.
So first, the nerve sends an action potential down the axon, and then the action potential opens up the calcium voltage gated channels, which then releases the calcium into the synaptic body, and then it binds to the vesicles, which have acetylcholine in it. And then the acetylcholine then releases through exocytosis, which uses ATP, and then it goes, the acetylcholine binds to the ligand-gated Na channel, and it puts in Na into the cell, and then potassium out. And then at the end of the signal, it uses the Na and calcium pump, so that uses ATP to bring them back where they started.
And then the action potential, after it crosses, keeps coming, and it goes into the T-tubule, which is this, and the action potential crosses into the sarcoplasmic reticulum through the voltage-gated calcium channels, and once it gets in here, that's what releases the calcium out into the cell.
So once the calcium is released from the sarcoplasmic reticulum, the actin has regulatory proteins called troponin and tryptomyosin. So the calcium will bind with the troponin, which opens up the binding sites on the actin, and then the tryptomyosin also opens up the binding sites. So then the binding sites will then bind with the ATP head of the myosin and go through the contraction process.
So then the head will cock and move the actin towards the myosin, towards the middle, shortening the sarcomere, and so look kind of like this. It will bind, and then cock and pull it towards it, and that uses ATP. So then it becomes ADP and P, and then another ATP will bind and will cock again and continue the process.
And then once the acetylcholine is gone, it'll go through relaxation, and enzymes will disintegrate or will break down the acetylcholine, and calcium will exit the cell.